Hello, I'm James Beasley. I'm a principal clinical scientist working from the West Midlands Regional Genetics Laboratory in Birmingham, England. And the title of my talk today is Maximizing Clinical Utility of a Rapid NGS Platform to Deliver FFPE and Liquid Biopsy Testing within an NHS Genomic Laboratory Hub. So here's the disclosure slide. Okay, so during this talk today, I'm going to begin by talking about the structure of genomic laboratory hubs in England. I'm then going to follow up by talking about solid cancer, specifically FFPE tests that are delivered in our lab and the challenges associated with delivering those tests. I'm then going to talk about the multi-panel NGS solution that's used within our laboratory and specifically how the rapid NGS amplicon-based technology that we use sits within that solution. And I'm also going to go through some provisional results from the rapid NGS testing of ctDNA, or in this case, ctTNA. Okay, so for those of you who aren't aware, genomic testing across England is divided up into 7 different geographical areas, with each area having a genomic laboratory hub, which acts as a lead contractor for genomic services across that region. And also within those regions, there'll be other genomic laboratories who are designated local genomic laboratories. So I work within the Central and South GLH, which is the purple area on the map, which is a consortium of 5 laboratories led by Birmingham Women's and Children's and serves a population of approximately 12 million. Within that GLH, there's Birmingham Women's and Children's Hospital, but there's also Oxford University Hospitals, University Hospital Southampton, Salisbury Foundation Trust, and University Hospitals Birmingham, who all act as LGLs. And the purpose of this structure is to deliver equitable access to genomic testing through the centralization and industrialization of genomic testing and to implement rapid, high-throughput NGS technologies. The genomic targets that must be delivered for each tumor type are defined within the National Genomics Test Directory, which is broadly divided into adult solid tumor, neurological, sarcoma, and pediatric. And I've just given a screenshot here of some of the tumor types that are included within the National Genomics Test Directory. So it's very broad. There are a large number of different tumor types that we have to deliver testing for. And the targets that we have to deliver include small nucleotide variants, copy number variants, and structural variants. There are obviously a number of challenges involved in delivering a high-throughput NGS service for acquired solid cancer. The first one being cost. So the chosen test needs to be affordable to the NHS because we're a publicly funded service. We need to generate results within a clinically relevant turnaround time, which essentially means we need to deliver results very quickly. We have to overcome the challenges of poor sample quality associated with FFPE. So we need to make sure that we can generate enough DNA and RNA, which is difficult given the sample size of biopsies, especially for non-small cell lung cancer. And we need to make sure that that DNA and RNA is of sufficient quality for next generation sequencing, which is impacted by various fixation protocols that are used by our different referral centers. We also need to deliver all of this testing at a very high throughput. So we need to meet the service demands whilst also building sufficient contingency in the service to consistently deliver high throughput, as we're the central provider for next generation sequencing across our region. So one way through which some of these challenges can be overcome is by making sure that we choose the right test. So we need to make sure that we choose and implement the right test at the right time in the sample pathway. So our approach within our laboratory is to deploy a multi-panel solution. So I've just used this diagram to illustrate how we've diversified our NGS portfolio over recent years. So in 2019, we were using a single 28-gene NGS panel. And since then, we've brought in and implemented numerous different NGS panels of varying size and chemistries across both DNA and RNA. And that means that the chosen panel can be tailored against sample quality, sample quantity, the targets required, the clinical urgency, and the clinical question being asked, whether that's for standard of care testing or for access to clinical trials. And this slide's actually out of date now, but we now have 7 different NGS panels which we can use for the different tumor types. So by having multiple panels available in-house, we can select the best test available based on the sample provided, process variable quality samples to provide a consistent service, be future-proof for additional biomarkers that may be approved, build on our already extensive experience in large NGS panels and develop our skills in using small NGS panels, deliver results in a clinically relevant turnaround time, provide the best value for money for our service users, and ultimately positively impact care and improve outcomes. So where does the rapid NGS platform sit within that solution? So we have two different rapid NGS panels in service, one 50 gene panel and one 163 gene panel. And I'll talk in a bit more detail about our experience with these two panels during this talk. But we've also got two other FFPE panels currently in development, a custom gene panel and a HRD panel. And at the same time, we're also evaluating the use of the 50 gene FFPE in its application for ctDNA, because the 50 gene panel is compatible with both FFPE and ctDNA. So the FFPE 50 gene panel can be run as a DNA only or a combined DNA and RNA panel. We've had this panel in service since November 2021 as a DNA only panel, and we introduced the RNA component into service in December 2022. We currently use this panel to deliver testing for lung, melanoma, colorectal, thyroid, breast, cholangiocarcinoma, and a handful of other rarer tumor types. And we found that the benefits of using this panel are varied. So the first benefit I want to talk about is that whilst the gene content of the 50 gene panel is focused on non-small cell lung cancer, the coverage is broad enough to be used for numerous other tumor types. So for example, because of the presence of KIT and PGGFRA on this panel, we can use it to test for GIST. For PIK3CA, we can test for breast. For KIT, NRAS, and BRAF, that makes it compatible with melanoma. NRAS, KRAS, BRAF for colorectal. Various different thyroid targets are present across the panel, dependent on the subtype that we're looking for. IDH and FGFR2 for cholangiocarcinoma, all of the essential targets for lung. And then also, because of the presence of FLT3, IDH1, IDH2, and TP53 on the panel, our hemato-oncology team have been able to use the 50 gene panel to deliver a rapid AML service from the hemato-oncology team. Another benefit of the panel is its high throughput. So because the panel is fully automated with an integrated bioinformatics secondary analysis solution provided, that means that samples can be sequenced, processed, and reported with high throughput. So to illustrate that, I just wanted to compare it against a hybridization-based 500 gene panel that we have in service. So for the 500 gene panel, the technical setup is 5 days. The sequencing time is 24 hours. And then that requires in-house bioinformatics support of around 4 hours per worksheet, with analysis taking approximately 30 minutes per case. Compared with the Amplicon 50 gene panel, the technical setup is just half a day. The sequencing time is 12 to 24 hours. There is no requirement for in-house bioinformatics support, and the analysis takes only 10 minutes per case. When that's applied to a normal working week, so our laboratory isn't currently a 7-day lab, you can see that from the 500 gene panel, we can begin setup on a Monday, and results will be available on the following Tuesday for 32 samples. During the same time period, it's very easy for us to set up 4 separate runs using the rapid NGS solution with a total sample process of 128. We've actually performed additional time in motion and mapping exercises that mean we can perform 4 runs during a working week per instrument that we have. So that's Monday to Friday. We currently have 3 instruments, which means that we can process a maximum of 384 samples per week, which is very, very high throughput for acquired FFPE NGS testing. The 50 gene Amplicon rapid NGS panel is also very tolerant of low quantity and quality samples. So again, just to highlight how tolerant it is, I've compared it against the hybridization 170 gene panel that we used to run in service. So for that 170 gene panel, the minimum input was 1 nanogram per microliter for a 50 nanogram total input. And compared to the 50 gene panel, we validated it down to just 2 nanogram total inputs. So that's a massive decrease in the minimum amount of DNA that we need in order to successfully test a sample. That meant that our pre-test failure rate, which means the amount of samples where we didn't get sufficient DNA or RNA for testing, went from 24.6% to 0%. And our post-test failure rate, which means that we obtained sufficient DNA and RNA for testing, but that DNA and RNA wasn't of sufficient quality to produce accurate results using NGS, went from 8.2% to 0.56%. So again, just to reiterate, before implementing the amplicon-based 50-gene panel, compared to after implementing the amplicon-based 50-gene panel, our pre-test went from 24% to 0%. Our post-test went from 8.2% to 0.56%. Our incomplete DNA or RNA, which means that we weren't able to generate results from all of the targets that were required to test, went from 36% to 7.9%. And our detection rate went up massively from 35.2% to 61.4%. The 50-gene amplicon panel is also very flexible in its run setup. So the panel is sequenced across a chip which has 4 lanes, and each lane can be loaded for a different application. So for example, across a single chip, we can use 2 lanes to sequence 16 paired DNA and RNA sample, the 3rd lane to sequence 8 DNA-only samples from FFPE, and the 4th lane we can test 8 DNA-only samples derived from blood and bone marrow for our rapid AML service, which moves me on to the application beyond FFPE. So as I mentioned, our hemato-oncology team are able to use the 50-gene NGS rapid NGS panel to deliver testing for TP53, IDH2, IDH1, FLT3, TKD. And the separate verification that was performed by a hemato-oncology team generated really good results when compared to their standard of care NGS panel. Okay, so now I'm going to talk a bit more about the actual performance of the panel in detecting variants. So that will be for SNVs, CNVs, and fusions, and I'll talk about each separately. But we evaluated the amplicon-based 50-gene panel for the detection of SNVs, CNVs, and rearrangements using a cohort of approximately 300 samples, so it was a very large verification. That sample cohort included clinical samples, EQA samples, and commercial controls, with comparator results generated via NGS, ddPCR, RT-PCR, and FISH. So I'll start off by talking about SNV performance. So a big part of us assessing the SNV performance of the 50-gene amplicon panel was by comparing results against GenQA samples, which is an external quality assurance provider, where they provided a reference result. So that was a result that we should be able to obtain with the result that we actually obtained from the rapid NGS testing. And all results were completely concordant, and we obtained full marks across all schemes. So that proves that the 50-gene panel is able to detect all variants that it's meant to detect. But we also wanted to assess whether the panel detected those variants at a variant allele frequency or a VAF that we expected. So we performed that assessment by testing some commercial controls where the expected VAF had been provided, and we mapped the observed VAF that we actually obtained from that panel against those. So the expected VAF is in blue, and the observed VAF is in orange. As you can see, the VAFs that we obtained were really concordant with those that were expected. And so this is data based on NGS, but we also performed the same assessment based on ddPCR, and again, the VAF obtained across the two are very, very close to one another. We also assessed CNV performance using this panel. And it's important to highlight that the copy number baseline is already built into the pipeline for the rapid 50-gene panel, which is quite unique compared to a lot of other NGS panels that are available where you have to sequence your own baseline. And that means that you can implement CNV testing really easily and really quickly when using this rapid NGS platform. This platform is also quite unique in that CNV calling is adjusted based on the tumor content of the sample that you're testing. So if you're testing a sample with a very low tumor content, the pipeline will adjust the copy number call upwards to accommodate for the contaminating wild-type DNA that's inevitably present in a low tumor content sample. So that theoretically means that the sensitivity for copy number calling is increased when using this panel, but it also means that it's really important that before you test your sample, you know you've got an accurate estimate of tumor content. Because, for example, if you look at this graph here, which shows the impact on copy number calling versus tumor content for individual samples, in this second sample here, if we'd been provided with a tumor content of 40%, the sample would have shown to be positive for met copy number, But if we were provided with a tumor content of 50%, they would have shown up to be negative. So it would have generated a false negative result. So it just highlights how important it is that you have the correct tumor content when using this panel. The main aspect of CNV validation that we performed was by comparing met copy number against generated from the rapid NGS platform against FISH, and all results were completely concordant, which was really positive. We have had one discordant result since going live with the service, where rapid NGS generated a negative result and FISH showed to be positive. And upon investigation, we realized that was because we were given an inaccurate tumor content percentage, which again highlights the importance of that value being accurate. So the final aspect of this panel that we verified was its performance in detecting gene fusions. So this panel can detect gene fusions in two different ways. It can either detect the gene fusion directly by sequencing across a breakpoint of two commonly known fusion partners, or alternatively, it can perform an expression imbalance calculation. So for the expression imbalance calculation, the software or the pipeline looks at the number of reads that have been obtained from one half of the gene compared to the reads that have been obtained on the second half of the gene. That's illustrated in this diagram here, where the first half of the gene has a low number of reads, but in the second half of the gene. So in the first half of the gene, there's a low number of reads, but in the second half of the gene, there's a high number of reads, which suggests that there's an expression imbalance event and that there's a fusion event between these 2 points within the gene. Now, the expression imbalance output from the software varies from the obvious. So in this one at the top, it's quite clear that there's a big expression imbalance occurring, to the more subtle in this graph at the bottom, where it's less obvious that there's a massive expression imbalance occurring. And these are both genuine fusion events. They've both been confirmed by an alternative RNA-seq platform. We've actually experienced a few issues with the expression imbalance output from the software, where there's a number of samples that have shown to be positive for an expression imbalance across various genes. So that's shown in this table here, where 5 samples are shown to have an expression imbalance. But we weren't able to confirm that result using an alternative RNA panel. And this is our very comprehensive RNA panel that we have available in-house. So we've determined that that is a result of a false positive on the rapid NGS platform. So we recommend that for fusion variants detected by expression imbalance alone, that they should be verified using an orthogonal method, and preferably an alternative comprehensive RNA panel before being reported. Okay, so in summary, for the amplicon-based 50-gene panel, it's fast, it's automated, it's very high throughput, it's tolerant, which means that no samples are left behind for testing. There's a BI solution provided, as well as a CNV baseline provided. It allows for flexible setup, broad application, and it has really good performance for SNV, CNV, and fusions. However, not all of the genes on the test directory are covered by the amplicon-based 50-gene panel, so it's not fit as a one-panel solution within an NHS laboratory. And we also say that fusion variants detected by expression imbalance alone should be verified using an orthogonal method. Okay, so I'm now going to talk about the 163-gene amplicon-based panel that we have available in our laboratory using the Rapid NGS service. And that's been in service since September 2023 as a DNA-only panel. Again, the 163-gene panel is an automated solution, and the content includes genes that must be tested in high throughput tumor types, which previously could only be delivered using our very large NGS panel. Again, copy number baseline is provided, which means that there's very little workup time required to implement new CNV services. The original scope of panel use, or the first phase of implementation, was for POLE testing in endometrial cancer, TERT promoter testing in thyroid cancer, and MDM2 copy number testing in liposarcoma. So I'm going to talk about each of those targets individually now and how they perform during the verification. So starting off with TERT, we tested a number of samples where we knew there was a variant present within the TERT promoter, and using the 163-gene panel, no results were concordant. So the 163-gene panel couldn't detect confidently the TERT promoter variants that we knew were present. So TERT promoter testing did not perform well in our hands, and that was driven mainly by very low coverage across the GC-rich TERT promoter region. So as illustrated in this graph here, it's very messy, but this is the coverage that we obtained for all of the samples during the verification and what percentage of each amplicon was covered to 350x. So as you can see, there's a lot of variability and lots of the amplicons and samples fall to 0 covered to 350x. So because of that, testing of the TERT promoter is not going to be delivered by us using this panel. However, performance of the POLE gene in endometrial cancer performed much better. Again, we ran samples where we knew the result, and all samples were concordant. Some amplicons across POLE did generate poor coverage. However, those regions were not essential for POLE in acquired endometrial setting. So we were able to bring the 163 gene panel into service to deliver POLE testing in endometrial cancer. So the final target that we wanted to look at was MDM2 copy number calling in liposarcoma. In order to assess this, we compared the 163 gene panel results with the results obtained from an alternative NGS panel and also FISH. And again, all cases were concordant. So across this graph are the copy number values that we obtained from the 163 gene panel versus an alternative NGS test, and all came back as positive. You might notice that the copy number calling for the dark blue, which is a 163 gene panel, is much higher than the light blue, which is the alternative panel. And that's likely because of the copy number variant adjustment that's being performed by this pipeline based on the tumor content of the sample that we tested. So to summarize, the 163 gene panel, again, it's fast, it's automated, which reduces manual errors, it's tolerant, a BI solution is provided, the CMV baseline is provided, it's a flexible setup, and it allows for a broad application. We did have poor performance across the TERT promoter region, which limited the scope of the initial validation phase, and it does have a lower throughput, the amplicon-based 50 gene panel, but it is much larger. So the next steps for the 163 gene panel are to extend the scope of the panel application to include other genes present on the panel, including BRCA1, BRCA2, MSH2, MSH6, MLH1, and PMS2. So now I'm going to talk more about some ongoing projects, and I'm going to start with ctDNA. So we're currently performing a verification on the use of the 50 gene panel to deliver ctDNA testing in-house. The expectations from a ctDNA NGS panel that we bring into service are that it must have a limit of detection of at least 0.5% VAF. It must have broad gene coverage to be used as a pan-cancer test. It must be able to detect SNVs, CNVs, and rearrangements. So the initial verification was performed using commercial controls, and we've also performed some additional verification using clinical research samples. I'm going to start by going through the results from the commercial controls. So we ran a variety of different commercial controls for ctDNA, one of which was a 5-gene multiplex control, and we ran that in 2 different formats. The first format in a 1% allele frequency, and the second in a 0.1% allele frequency. Within this graph, in green is the expected allele frequency that the variant was meant to be found at, generally around 1%. And then the variant allele frequency that was obtained from the rapid NGS result in dark blue, with light gray and light blue showing the variant allele frequency for 2 alternative ctDNA panels that we tested for comparison. So for the 1% control, the rapid NGS panel was able to detect all variants around the variant allele frequency that we were expecting. However, for the 0.1% control, the rapid NGS panel was only able to detect some of the variants present within the control. So that led us to believe that the limit of detection for the rapid NGS panel was somewhere between 0.1 and 1% allele frequency. So to define that a bit further, we also tested a 0.5% variant allele frequency control, and we actually ran these in triplicate on the rapid NGS panel, so the first 3 bars for each variant, and the rapid NGS panel was able to detect all variants present within this control. So that led us to believe that the rapid NGS limit detection sits somewhere between 0.1 and 0.5%, which means that it meets the minimum limited detection requirements for a ctDNA panel. So whilst we've proven that the rapid NGS panel has sufficient LOD when detecting SNVs, the next step was for us to prove that it can detect copy number variants and structural variants. There are very limited commercial controls available which contain these variants. However, we now have a pool of clinical research plasma samples where SNV, CNV, and SV structural variants have been detected in tissue. And we've already been able to run some of these samples which have shown good results. So I've got the results here from 4 of those samples. So for sample 1, 2, 3, and 4, this is the expected result that was obtained from tissue in the first column. And this is a result that we actually obtained from the rapid NGS platform. So for sample 1 and sample 3, we detected SNVs in tissue, in a KRAS and an EGFR, and the rapid NGS platform detected those same variants in plasma. So that's full concordance with SNV. However, for samples 2 and samples 4, an ALK fusion was shown to be present on the tissue, but we weren't able to detect any evidence of that fusion when testing the plasma. So that could be because the platform isn't able to detect gene fusions in ctDNA, or there could be a biological reason why the gene fusion is no longer present within the plasma sample. And that could be because the blood was drawn post-treatment with ALK inhibitor. But we just need to define that process and define the pathway and how far along in the treatment cycle these samples were when they were taken. So that requires a lot more investigation from our end. So finally, I just want to talk about a future project that we have ongoing using the rapid NGS platform, and that is a custom research BRCAness panel. So this is going to be a focused panel to deliver testing primarily for prostate, endometrial, and breast cancer. And the gene content includes BRCA1, BRCA2, PIK3CA, POLE, ATM, and CDK12. And using this panel, 24 samples can be tested across a single chip, across 2 runs. And it has a much increased throughput compared to the 163 gene panel. We've only run 12 samples so far, but we're really optimistic about results, and we're going to continue on with the validation of this panel and hopefully bring it into clinical service soon. Okay, so that's everything I wanted to talk about today. Thank you for listening, and here's my email address if you want to get in touch about anything that we've been working on.